Welcome and thanks for joining us. My guest today is Dr. Catherine Jennings. She's the lead scientist at the Applied Science Research Center and senior manager for the life sciences team at Nobilis. Dr. Jennings, good to have you with us.
Thank you so much for having me.
And I wanted to ask you basically here that in the area of biosurveillance and biodetection in the natural disaster and widespread sense, there were really some important lessons learned and some changes probably the nation needs to implement following our learnings in the pandemic. Would you say that's a fair way to assess it? And tell us more.
I do think that's a fair statement. I think there was a lot of good that we learned and we're implementing in the post-pandemic era. I think there are some things that we can do better. On my team, as far as biosurveillance goes, we're particularly focused on molecular-based biosurveillance and looking at whole genome sequencing and bioinformatics analysis that goes with that.
Right. So let's also place this in the pantheon of that whole Siberny type of endeavor: chemical, biological, radiological, nuclear, and explosive. Biological is unique. I mean, there's not really much all of those disciplines have in common other than that they need to be surveilled, correct?
That is correct. And one of the unique things about the bioagents is that they proliferate. So over time, you can have a bacterium or virus increase in number, and that makes it more difficult to control than, say, a chemical agent.
All right. Well, you have done a couple of studies there at Nobilis, some published work here that really lent some light onto all of this. One has to do with monitoring molecular diagnostic test performance. And really, that's, I guess, in the post-pandemic era, as vaccines come in, that becomes really important. Tell us more about that.
Yes, thank you. So this work started in the pre-pandemic era, where we were basically monitoring the performance of PCR assays, so polymerase chain reaction assays that are very popular molecular-based tests for the presence of an infectious agent in somebody's clinical sample. So to give you a visual image, in a polymerase chain reaction, if you have one genome copy, so if you think of DNA, for example, as a ladder and it has rungs between the supports, if you are in a PCR reaction, you heat that up, the ladder comes apart, and now you have two templates.
All right, so you have two ladders, and what can you do with two ladders that you could not do with one ladder?
So with two ladders, it's easier for us to detect the target gene sequence. And using, say, 30 cycles of the PCR reaction, we can go from 2 to billions. And so you can imagine how that's so much easier to find your gene target, especially in a background. So PCR is popular because it's sensitive and specific.
And give us the practical effect of this technology, this polymerase discovery with the two ladders and the billions of samples. What does that mean for biosurveillance?
So for biosurveillance, if you take a human sample, you can imagine that the pathogen or the disease-causing agent is going to be in a much lower copy number than, say, the human DNA that comes out with the sample. So we want to be able to amplify the disease-causing agent so we can detect it. And we can say, this person has, for example, a SARS-CoV-2 virus infection versus something else. And we can detect what's causing them to be sick. And it's a very rapid method. So this has really revolutionized disease detection in a lot of ways because it is sensitive, specific, and rapid.
And therefore, if something happens to someone and that cause can be determined more quickly, then that means other people can be tested and the whole process of detection then speeds up?
Yes, that's exactly right. One thing we worry about, one of the challenges is, for example, with SARS-CoV-2, we know it was mutating over time. And our concern is that those mutations might happen in the target regions that we're looking for with PCR. So our paper was about monitoring 43 different PCR assays for SARS-CoV-2. As more and more genome sequences were becoming available, we were able to look for mutations occurring that would cause those PCR assays to fail. and then we might lose the ability to detect that infection in someone. So it's a really, our method is really based on using whole genome sequences and monitoring for signature erosion to keep those PCR diagnostics as fresh and as up to speed with the virus changes as possible.
So as the COVID itself mutates and takes on new forms with new and exotic names and different infection waves, it sounds like those that try to keep on top of what's going on, let's say CDC or other elements of Health and Human Services, then have tools that can make the identification of the variants much more apparent much sooner.
Yes, that's correct. And so if we're using whole genome sequencing in addition to PCR, we're able to characterize what those mutations are, where they're occurring, and what they mean in the biology of the virus, especially as it relates to causing disease. So it's more than about just detection. It's also about characterizing that agent.
Right, because you're seeing right into the molecular makeup of it, in other words.
Correct.
And we've got a couple of other things going on. There's like a monkeypox scare now, and that's beginning to build, I think, in the public mind and also in terms of actual cases occurring. And some of the older virus types of outbreaks haven't really disappeared. Maybe they're less common in the United States, but they are worrisome around the world. I'm thinking of Ebola. And so, can this polymerase type of technique that you've described help with all types of virus and not just the COVID?
That's correct. It can be used for all types of viruses and is commonly used. We have recently designed a set of monkeypox primers, and we're in the process right now of characterizing how effective and specific they are. Uh, the PCR can be used for any infectious agent. So it could be bacteria, anything that has gene sequences. And it's been
But
used for quite many years.
because the Ebola and especially the COVID type of virus is the COVID is a virus that was already understood in the general type of virus that it was even before COVID-19 came out. But Dr. Fauci and some other federal officials and some other doctors have noted there are many, many more types of viri that have not caused these types of outbreaks yet in the human population, but we should probably worry about them. So would this have wider application beyond the COVID type of virus, which has manifested itself, other ways earlier than the current outbreak?
Right. Yes. So the family of viruses that the coronavirus that caused COVID belongs to is a very large family, and there are many viruses within it. Some cause human disease that are very mild, like just a cold, and then there's others like SARS-CoV-2. We know now has caused a significant respiratory infection. But in the viral and biological world in general, there are many more organisms and agents that don't cause human disease than do. But one thing you hear about in this field is zoonotic disease agents. So those are viruses and bacteria, for example, that cause disease in animals, but can also cause disease in humans. And what we're very concerned about in this field is how, as human populations travel more, encroach on wildlife habitat, live more tightly packed in urban areas, we're really changing that dynamic of interaction with animals and their pathogens. So we're seeing this increased number in zoonotic diseases growing because we're in more contact with those viruses and bacteria than we used to be.
And I guess for people that worry about animal populations and wildlife, it works in reverse, that humans can give certain wildlife species a real big problem that causes their numbers to drop. And I know Fish and Wildlife Service, elements of the Interior Department and agriculture worry about the reverse happening. So is the technique you've outlined also useful for, say, federal surveillance in livestock and in, say, protected species?
Yes, it has lots of applications for those very things. And one thing that I'm interested in terms of biosurveillance and seeing it grow is to surveil the environment with a lot more systematic approach and just at a greater scale. So we better understand what's in our environment and what we're going to come into contact with, which may help us push out that prediction step and the early detection beyond what we saw with the pandemic. We were just too late to the game to really intervene and control it earlier. But with more broad surveillance, I think we might be able to do a better job.
And so translate that for us into practice, say by those that do surveillance on behalf of the government and its partners. There are healthcare networks that also do this. What would that mean in terms of practice of biosurveillance so that we don't wait till people are dying and wake up and say, oh my gosh, something's going on? It sounds like there's a predictive quality that could be brought in here that was not evident before.
I think that's true. I think there is a predictive quality. It's challenging. It's dependent on mathematical models. And in some ways, we just need a lot more data for those mathematical models to be more accurate. So I see one of the big things that we as a community need to do is to gather more data so we have those reference databases and our models become more accurate.
And by the way, what did it take to develop this methodology of making the two ladders and DNA sequencing and all of that? It sounds like something that you can't quite do with an erector set.
You can't. So that actually is probably a little bit outside of the details I remember. It started back in the 70s. One of the pioneers in the field got a Nobel Prize for it because it has been such a game changer for this field to understand that we can manipulate DNA in a way so we can make lots of copies and start to see what was unseeable before.
And I guess I was going to focus a little bit closer in on Nobilis activity with respect to this type of technique, because you are the Applied Science Research Center lead scientist there. And so what types of activities happen there that get you up to speed on these technologies?
So we do a couple of things. One is that we are very interested in whole genome sequencing, but in particular, building out the bioinformatics analysis that goes with it. So this is something we saw during the COVID pandemic, is that there is a need to build out those informatics tools and also to automate them, make them easier to use and more informative in terms of interpreting what the data look like. So we spend a lot of time not only ingesting the raw sequence data, but making those bioinformatics algorithms and tools, visualization tools, and trying to make them easier to use so we can make biosurveillance more accessible to more populations, to push it out to the field for animal surveillance and environmental surveillance and healthcare workers.
Right. So the more that the technique produces large data sets, and it does produce large data sets, order of magnitude larger than older ways, then it becomes a data handling, data analysis, algorithmic type of exercise and information technology as much as a biotechnology, sounds like.
Absolutely. And that's one of the interesting things I think in biology today is that it is becoming more data dense. And we are reliant more on computer scientists and mathematicians and engineers to bring into this biology field and really help us make sense of all the data. And that makes it exciting. And it's certainly a field in evolution and rapid advancing, especially on the data end.
Some fascinating insights, but that's not all. We've got much more after the break. My guest today is Catherine Jennings. She's the lead scientist at the Applied Science Research Center and senior manager for the life sciences team at Nobilis. I'm Tom Temin. This discussion is Evolving Biosurveillance in the Post-Pandemic Era, sponsored by Nobilis here on Federal News Network.